How is everybody doing? Good? Had a good weekend? Yeah, ready for the test? Okay. Well, this is how your testing pairs is going to work. Okay, just keeping in the back of your mind a bit of overview. I hope you have chosen your testing buddies by now, right? If you want to take the test by yourself, it's fine too, okay? If you change your mind in the last minute and you want to find another lonely test taker, it's okay too, as long as you don't disturb the class too much, okay? Now, for the first 45 minutes or so of the test, of course, you're going to come with your own Scantron, right? And each one of you is going to have a test paper, right? So for the first 45 minutes or so, I ask you to be like as quiet as possible. Just confirm, like you're cheating, like look at the answer, kind of confirm your answers, and mark the ones that you have questions. And then for the last half an hour or so, then I want you to just whisper, like it's still like you're cheating, okay? So it's like open cheating, but only with your, designated test partner. Okay, so this way we can just keep a very, very quiet environment, and you just pretend you're cheating is a bit more exciting, but then you have permission to cheat, kind of that kind of thing. What we don't want to happen is a lot of conversation, and then that thing can distract everybody else, and that's going to defeat the purpose. So pretend you are professional cheaters, kind of. Okay, so that's the whole idea for the exam. Okay, before I go into MHC, do we have questions, like burning questions, or whatever I covered? Thing? I guess this is like when you say we have questions, this is when everything goes blank, right? Yeah. Okay.
I wanted to ask, there's different kinds of effectors, right?
Yes.
So with the complement activation, is that the same thing as degranulation?
You mean your antibodies?
Yeah.
Okay, effectors of antibodies. So that's a good question. You have basically 5 effector functions of antibodies, right? And then you have your, you mean agglutination and degranulation?
Mm-hmm.
Yeah. So agglutination
is when they come together.
Is when you have a blob that gets formed. Mm-hmm. So you have one antibody that will bind, for example, to one red blood cell, mm-hmm. and then the other arm of the antibody will bind to another red blood cell, mm-hmm. and then you have another antibody that is binding to this red blood cell, but to a third red blood cell, mm-hmm. and those will start to really clump up and make this blob that can clog like a blood vessel. Okay. When you talk about degranulation, that's a different thing. That is a function of an antibody, like for example, IgE, that can bind to a parasite, and then the constant region of that antibody is what is going to connect to a mast cell or to an eosinophil that is going to get activated once that antibody is bound to the parasite. So that other white blood cell is going to dump toxic stuff on the surface of the parasite. So that is called degranulation. Okay, so that's the difference. So agglutination is blob formation. Degranulation is when a white blood cell dumps toxic stuff on the surface of some pathogen.
So what are the factors? Neutralization, oxidation, AD60, agglutination, and
there is degranulation, and there is also precipitation. That will be 6. I'll keep it to the 5. It's fine. Okay. No, precipitation is very similar to agglutination, but it's okay. Forget it. Okay, we keep it simple. Okay. All right. So this class here on MHC molecules is actually a fun class, and is a class that is kind of like intuitive once you get in the mood of how things are happening. Okay. And yes, I did some changes just to make it flow a little bit better and not to be too repetitive, like in bits and pieces all over the place. Okay, so here are your objectives. You can read that on your own. So here we start with the overview, right? So we know that the T cells are not going to recognize native proteins in their 3D conformations, right? Of course, there are always exceptions, but most of the T cells are not going to be able to recognize 3D, right? So they have to see a little piece of stretched out protein that is served on this MHC platter, right? So this is the 2D version, 2D view of a T cell, right? That means the peptide to be presented has to be processed and has to be stuck on the MHC in order to be properly presented to the T cell that's going to recognize. So this is when we start talking about these MHC complexes, right? So major histocompatibility complex, and then you should be thinking, if there is a major, is there a minor? Yeah, there are some minor ones, but we just stick to the major. Okay, so now we know that these are complexes of genes that are inherited, right? So at the same time, they are genes that are not getting mutated. You don't get recombination of those genes, okay? So that in the back of your head should start bringing some red flags, because remember all the recombination that goes on for the T cell receptor and for the B cell receptor, because they actually have to recognize all these different types of antigens out there, right? Now, when you talk about T cell receptor recognizing antigens, all the antigens are going to be presented on these MHC molecules. So how can the MHC molecule, without all of that somatic combination, how can it present all different types of proteins, since in this like finite number of presenting cell or molecules, right? So that's one of the things that at first is a bit is not very intuitive. It kind of gets a little short circuit in the brain, okay? But this is what happens. You have a limited number of genes that are made to present certain peptides to T cells, okay? Now, we know that when the MHC molecules are not matched in case of transplants, we know that we get rejection of transplants, right? And this is really how all these studies about MHC molecules came about, right? On rejection of transplanted grafts. But then, of course, these molecules did not quite evolve first to study rejection of grafts, right? But this is how we started to learn about those. So the human genes for MHC, they're called human HLA, leukocyte adhesion molecules or something like that. So the genes are HLA. They're called HLA. So when you read the books, you're going to find different names for the genes and for the molecules, because a lot of people were studying the mouse, and then some other people were studying humans. They didn't really know they were studying the same molecules, so then the guys who were studying the mouse gave one name, the guys who were studying the humans gave another name, and now we have to deal with both names, one of those things. All right, so we stick to the humans versions of things. All right. So we know we have two classes of MHC. You have the class 1, that is going to be presenting a peptide to what kind of T cell?
CD8.
CD8 or cytotoxic T cell, right? Now, the MHC class 1 comes from HLA genes, remember those are humans, right? The HLA genes that are actually 3 different genes, okay? So you have 3 different molecules of MHC class 1 that are inherited in 3 different genes, and then you also inherit 2 chromosomes, right? 1 from each parent. So you're going to be expressing a lot of those genes. So the genes that are making MHC class 1, those are genes HLA-A, HLA-B, and HLA-C. Okay, and you're going to see more about those. Okay. Where do you find MHC class 1? On all nucleated cells in the body. So if the cell has a nucleus, it's going to have MHC class 1. Which one is the only cell that you do not have a nucleus and you do not have MHC class 1? Red blood cell, exactly. Is that a test question? Oh, of course. Okay, so here. And here you have the second class. That is, of course, the class 2. The genes that are coding for class 2 are HLA, DP, DQ, DR. And I always have a little trouble remembering, why D? Well, because here you have A, B, C. Next one is D. Okay. So the double letters for MHC class 2 is going to be DQ, DP, DQ, DR, okay? And you can associate that class 2 has 2 letters. Okay, so you know that those 2 letters of the genes are related to the class 2. Now, only a few types of cells are going to have MHC class 2, and those are going to be your antigen-presenting cells, professional or not. Of course, dendritic cells are the real professional ones, right? So that means your macrophages as well as your B cells are going to have MHC class 2. And of course, you have to have those thymic epithelial cells expressing MHC class 2 because they are involved in teaching your thymocytes about positive selection and then negative selection. Right? So they have to express those MHC class 2. And then what are those molecules doing? They're going to interact with your T helper cell or CD4 positive cells. Now, besides those types of antigen presenting classes, you also have other types of cells that can present MHC class 2 if they are under some type of inflammation process. That can be like epithelial cells or some endothelial cells or some cells, I think in the liver or somewhere. There are some specific other cells that can get MHC class 2 to be expressed, but not really all the time. Okay. So, which one is the chromosome that gets you to inherit those MHCs? Well, that's chromosome 6. And remember, you're getting one from your mom and one from your dad. Right. So when you look at the stretch of chromosome 6, which I do have a picture showing the entire chromosome, you're going to find your genes for MHC class 2. So those are 2 genes that are close together, and this is going to make here your HLA-DP, HLA-DQ, HLA-DR. Okay. So each one of those gene segments is going to make a peptide. And then those 2 peptides are going to associate and make your MHC molecule. Okay, so you get 3 genes of those from your dad, 3 genes of those from your mom. And then when you look here at your MHC class 1, you're going to have 1 gene that is coming on this chromosome. And this gene is going to make the bulk of your molecule that is going to be embedded on the membrane. Okay, but you're also going to have another gene that is coming from somewhere else that is going to associate with this part here of that molecule. Okay, so you have this 1 gene that is making this 1 long molecule, but if it just goes like that, it's not stable. So you're going to have another beta chain that is going to associate that with that and really make it stable, which you're going to see next. So here again, all nucleated cells have MHC class 1, and we know that the job of MHC class 1 is to present a piece of a protein that was present in the cytoplasm and that got chopped up and is going to end up on the surface of the cell. Okay, MHC class 1 is always associated with an antigen, which is a piece of protein, right, on the surface of the cell. Always associated with an antigen. Okay, so basically what that is doing is showing to the T cells, to the cytotoxic T cells, is showing the health states of those cells. Even platelets, they have MHC1, and platelets don't have nuclei, right? They're just cell fragments, right? But basically the cells, they are going to always be showing a piece of the protein to cytotoxic T cells because the cytotoxic T cells are really keeping track of the health of your body cells, okay? So if the cytotoxic T cell goes there and doesn't recognize that that protein, then everything is fine, right? Because this is what does the job of the cytotoxic T cell. It's just to make sure everything is fine, right? And why is it that your cytotoxic T cell wouldn't recognize a piece of antigen in MHC when everything's okay? Okay, why is it that your cytotoxic T cell doesn't go there and kind of kill the cell? Okay, we'll get there again. Let's keep that in the back of your mind, okay? I think I'm just going ahead of myself here, but that's an important question, okay? All right, so let's see what happens here. When you look here at MHC class 2, again, you have your antigen-presenting cells, right? And you also have non-antigen-presenting cells that can have inducible expression of MHC class 2. And here are your cells. You have endothelial cells. You have, of course, your thymic epithelial cells, and then you have some fibroblasts and some epithelial cells. That's like in your on your skin, okay? And under inflammation, those two types of cells, they can upregulate MHC class 2. Okay, so here, let's start looking at how that process works, okay? So you have, in this case, your MHC class 1, which of course is not really drawn to whatever it's supposed to look like. It's just a box, just a square, right? So you're going to present a piece of peptide. One thing that is good about this representation here is that you see that this little piece of peptide is inside of this structure over there. So the piece of peptide is going to be presented there is actually quite small, okay? So what's going to happen here? Your T cell receptor is going to recognize that, but of course it's not going to be able to do that if you don't have interaction of your CD8 molecule with a part of your MHC class 1 that is actually quite stable across all different alleles, okay? So CD8 is going to interact with the part that is not very variable in allele expression in the population. Okay, so what's going to happen here? If this thing recognizes, this cytotoxic T cell is going to either bind to receptor on the cell surface, that is the Fas-Fas ligand interaction, and is going to tell this cell to commit suicide, or the T cell is going to dump some toxic stuff called perforin and granzyme and punch holes on the membrane. But those mechanisms we're going to see into more details later. Okay, so now we're really focusing on MHC interaction. All right, so here. When you look at MHC class 2, they kind of look like the same kind of square, right? But this is not very accurate because this little peptide on MHC class 2, it can actually go across those binding sites here like a hot dog on a bun. Okay, so it's not totally limited to the inside of that groove. Okay, so when you have interaction of MHC class 2 to the helper T cell, what the helper T cell is going to do, it's going to start releasing some chemicals like cytokines that is going to help the development of other cells and send specific signals like for B cells, right? So basically the helper T cell is going to produce chemicals to tell other cells what to do. Or you could also have a helper T cell that is a regulatory T cell, and that one is still going to produce chemicals to get everybody quiet, which is still telling them what to do, right? So this is different than the killing action of your cytotoxic T cells. Yet the CD4 molecule is also going to interact with a non-variant part of your MHC class 2, right? The variant part of all the alleles, that variant part is really going to be on this binding site here, okay, for both of those molecules. Okay, so here is your chromosome 6. Okay? So here's the centromere. This is the long C arm, and this is its shorter P arm. And here are the MHC locus for the genes. Okay, so you're going to have HLA type 1, HLA type 2, HLA type 3. Well, HLA type 3 is not really going to code for MHC molecules, but they do code for other types of proteins that are involved in this process. Okay, so we're mainly focusing on the HLA1 and HLA2. Okay. Now, let's look at these different sessions here. Those proteins, they are expressed in a manner that is co-dominant. Everybody took genetics? Yeah, you remember what co-dominance means? They are expressed and both of them have a say, right? One of them is not going to mask the expression of the other. They're both going to be expressed and having a say on the expression and their function, okay? Now, those genes are very, very close together, okay? So usually they are shifted, like they are inherited, like all at once with the chromosome. They never really shuffle from one chromosome to another. Like they tend to not get mixed up. Okay, so this property of getting inherited as a bunch of genes that are going together, that is what you call a haplotype inheritance. Okay, so you're going to inherit one chromosome 6 from your mom and the other one from your dad, and all of those genes are going to be expressed in a co-dominant form in every one of your cells, okay, that have the expression for those genes. Okay, so here, let's see more details here. So here is this idea of this haplotype inheritance, right? So haplotypes mean that you're just inheriting the block of genes, okay? And since you have co-dominant expression, let's see what happens here. When you look at the genes that you can inherit from your mom, you have certain alleles that mom has, right? So she has actually, let's say here, 2 alleles of this type, 2 alleles of those types, which are really 6, 12, 24. It's a lot of alleles that you're inheriting, right? Remember, this one here is going to produce 3 proteins. This one is going to produce 3 proteins, right? And then you're going to look here at your chromosome 6 from your dad. So basically, what are the possible combinations that you can get of chromosome 6 from mom and dad, right? So you see that they are independent terms. So you multiply independent terms and you can come up with 4 possible different combinations of inheritance, okay? That tells you that most likely your siblings are going to have a different combination than you have of this inheritance, of haplotype inheritance, okay? So you basically have, on average, 1 in 4 siblings that is going to have the same type of inheritance of these haplotypes. Well, because you can have 4 possible combinations, right? And this is why when you, for example, are looking for organ donor, like a kidney donor, and the first thing you look is at, whether your siblings have the matching haplotypes, right, or not, you basically have only a 25% chance of having a match, right? So most likely you don't have a match. And then if you look at the parents, then you for sure don't have a match because you only have one of those chromosomes from the parents, so you have like 50% difference, right? So it's even like worse. Okay, so it's very, very difficult to get the same genes that are inherited for matching of those MHCs. Okay. And here is even a situation that is very rare in case of recombination of those genes, right? The reason why this is very rare is because those genes are so close together that you don't really have a lot of space to allow this type of recombination. Remember, you have those like holiday crossover vacations, I mean, not vacation, I mean, type of formation or something like that. So because these genes are so stuck together, you don't have enough chromosomal space or enough, template space to have all those things going on. But sometimes it can happen. And then in that case, you have total non-matching of your siblings, right? Because I mean, if you need a transplant, you're doomed, right? Okay, then you have to really look in the whole population and see if you can get a match. But then situations even get worse. Okay, so these are the non-matches. Now, one question is that we know that the MHC alleles are the alleles that have the most number of different alleles in the population. You have like so many different alleles. Okay, there's no recombination here. It's just very, very much different alleles. And then the question is, why is it that the population in general has so many different alleles for these few genes? Okay, so some reason is that when you look at the groove of those MHCs, so let's say, for example, the MHC class 2, right, that you know you're going to present exogenous peptides. When you look at the groove, you have certain residues on the groove that allow for binding of certain pieces of proteins that are from, let's say, a virus like coronavirus, our favorite lately, right? So if you have one population of alleles that can really bind peptides from coronavirus and totally activate your T helper cells and your adaptive immune system, then the population who inherit those genes, they're most likely going to do well in case of a pandemic. Okay. If you have a certain population that doesn't really recognize binding of those proteins, most likely that population is going to die because they don't have enough defense against the virus. Okay, so you have once in a while you have this sweeping of certain alleles just because we have pandemics that happen, right? And some MHCs are better than others to really keep the species alive. Okay. And of course, there are many other factors that are related to immunology and keeping you alive in case of disease, right? But this is one of them, okay? So pathogen identification is one of the reasons why you have so many different types of alleles. Hopefully, somebody in the human population is going to have that one allele that is going to be very resistant to dying from a certain pathogen. So then the species can survive, right? Another reason is that you also need to have a huge variety in your T cell receptors, right? So having different MHCs is going to broaden your T cell receptors. About the same idea on the defense of pathogens. Okay. Now, another thing that is interesting is that depending on the allele that you have for the MHC, which we are not really going to get into those details in this class because those are more for pathology and for medical school level immunology, is that certain alleles make you more susceptible to developing some types of autoimmune diseases, including type 1 diabetes, Crohn's disease, and some other types of autoimmune diseases, lupus, erythematosus, and all these other types of autoimmune diseases that depending on the MHC molecule or the MHC allele that you have, you really have a lot of more chances of developing that throughout your lifetime. There is one disease that is a problem of the spinal column, ankylosing spondylitis, that not only you have the MHC allele that is very prominent for that, you also have a linkage to being male. So that's kind of strange. But when you start looking at the genetics of these autoimmune diseases, it's really, really fascinating. Okay, so if you have a huge variety of alleles in the population, most likely you are going to to mate with somebody that doesn't have that same problem. So most likely your offspring is going to have a better chance at surviving the tough world out there, right? And here is when I have this little observation on the T-shirt experiment. Anybody heard of this T-shirt experiment? Anybody know what I'm talking about? No? Okay, so when you look at other species of mammals, we know that MHC molecules trigger their attractiveness to the opposite sex, okay? So some immunologists came with the idea saying that, wait, we are mammals. You know what I'm talking about? I mean, we are mammals. Is it possible that we're also attracted to somebody else's MHCs. They are very different from our own. You know, so this is what they did. Okay, they got some people at the gym. They got some girls, you know, some guys. Of course, college students, they always volunteer for, you know, for some experiments, right? So they're working out at the gym, you know. So then they donated. They got a very sweaty T-shirt, very attractive sweaty T-shirt. Yeah. And so, and they gave it to the, you know, to the scientists. And then there was another room of other college students and volunteers. Their task was to smell the T-shirt and tell the researcher which T-shirt they were attracted to the most. Yeah. So they had no idea who wore the T-shirt, right? It so happens that the people who were smelling the T-shirts, they were attracted to the person who has the most different MHC genes than their own genes. So now you're like, how is that possible? Right? I mean, what's going on here? How can you smell MHC? Right? I mean, when you are exercising, you are under a certain level of inflammation. I mean, it's a healthy level of inflammation, but remember, your epithelial cells are also going to produce those MHCs. Somehow, people can smell it. Okay? And it's interesting that, you know, you say like opposite attract. In this case, it's like very different MHCs are very attractive. Okay, so that means that, you know, something is happening in the back of the brain telling you that go with a person with the most different MHC and your offspring is most likely going to survive some pandemic. All right, very, very clever thing to do, right? So that's the story, the overall story with the MHC. So now look at the structure of the MHCs. Okay, that would be different. I get your 23andMe, and before you know about the composition of the persons, let's smell the T-shirts. Oh, okay, just look at the genetic code and to see. Okay, so here, when you look at the structures, right? So you look here at your MHC class 1. How do you identify it right away that this guy is MHC class 1? Well, he has only one leg stuck to the membrane. Okay, so it's one transmembrane, and is also only one polypeptide that is connected. So this polypeptide, it has domains that are alpha 1, alpha 2, and alpha 3. Okay, it's one long polypeptide folded in this way. Now, it needs a support, otherwise it will collapse on the surface of the membrane. So the support comes from this other associated protein that is not covalently bonded, is only associated. Okay, this protein is called beta microglobulin, and this is the guy that is coming from a different chromosome. Okay, but is constitutively expressed, is very much non-variant, meaning that everybody has the same allele for this guy. There is no difference in alleles here. Everybody has beta microglobulin. Okay, so this is the overall structure of that. And when you look at this groove here, you have this groove between alpha 1 and alpha 2, and this is where your little peptide is going to bind. Okay, now when you look here at your MHC class 2 structure, you're going to have 2 membrane spanning domains. You also have 2 polypeptide chains. Okay, so one of them is alpha 1, alpha 2. The other one is beta 1, beta 2. Your binding groove is between those 2 chains, meaning between alpha 1 and beta 1. That's when you have your little binding for your bigger polypeptide. Now keep in mind that this groove here is smaller than that groove over there. Okay, so the piece of peptide that goes there is going to be bigger than the piece of peptide that goes here. Okay, so let me see here. Okay, so this slide is important to show you that the polymorphic regions of all of those alleles, they really have to do only with the binding session of both class 1 and class 2. Okay, so when you look here at class 1, this very polymorphic region is going to be between alpha 1 and alpha 2. When you look at your class 2, the polymorphic is going to be between alpha 1 and beta 1. Okay, the rest of the protein, you're going to have those non-polymorphic regions, and those are important because they're going to interact with those either CD8 or CD4 molecules. Okay, so it's the same story with both classes. All right, so here a bit more on the number of possible alleles and possible combinations that you can have. Okay, so this slide is showing that this is chromosome 6 that you're inheriting from your mom. Okay, each one of those genes, is this for MHC1 or 2? Those genes are coding for MHC1 or MHC2? One, it has only one letter, right? ABC, that is MHC1. So here you inherit from your mom, and then those here you're inheriting from your dad. Now you say, what are those numbers? Those numbers here, 8, 17, 84, those numbers are just to indicate which allele you are inheriting, because there are so many alleles in the population. Like when you talk about the alpha chain, right, of your MHC1, you have over 4,000 different alleles for your B gene. You have over 2,000 different alleles for C, and then over 3,000 different alleles for A. I'm getting those numbers from here, okay? So that means this number here, 8, was like number 8 out of 4,000 possible alleles, okay? So these are the numbers means. Now, out of these genes, you're going to get protein expression. So on the surface of your cell, you're going to have this, and that is being expressed. So 3 different types of MHC1. And now here you put the MHC1 genes from your dad. So dad had given you B27, C46, and A2. So you look at the surface of the cell, and you're going to have 6 different types of MHC class 1 that is expressed. Okay. So looking at your MHC class 1, you're going to have here, where is MHC? Right here. You're going to have all these possible combinations of alleles for your alpha chain. But then when you look at your beta microglobulin, you only have one option for allele because everybody has the same. Okay? Again, this is exemplifying what codominance means. Okay, and then the same story or similar story happens to your MHC class 2, that you're still going to have 6 different expressed proteins. You put them all together, you have 12 different proteins that can be expressed on a cell that is expressing both types, and they are codominant. Okay. All right. All right, so here are the ones from your mom, and then those are the ones from your dad, and everybody gets expressed. All right. So looking a bit more on the structure of your MHC class 1, remember this is one long polypeptide. This is your beta globulin non-variant chain, right, is associated. And then here you have alpha 1, alpha 2, alpha 3. And right here in the middle of alpha 1 and alpha 2, you're going to have the little groove for your antigen. Most of the variation of the alleles is going to happen on the secondary structure that is right on the base between alpha 1 and alpha 2. And yeah, that is a beta sheet. Yeah, sorry. Yeah, confusing. But yeah, I'm not going to ask you about that. But know that the variance is going to be right there on this plate here of beta sheet that is on the alpha protein. Okay. And remember also you have one anchor to the membrane. All right. So here is a simplified model of your groove and the domains of the protein. Okay. So here you have the plate of your beta sheet, and keep in mind here that the edges are closed. So your protein really has to sit inside of that little mouth, kind of looks like a mouth. So here's a presentation for your class 2. So remember, you have 2 anchoring portions, right? So 2 different proteins, and your binding groove is going to be between alpha 1 and beta 1 right there. And I don't know what happened to my between alpha 1 and beta 1. And then let me see, this is again is going to express, is going to put a piece of exogenous peptide, right? And those peptides are going to be longer than the ones you can put on MHC class 1. So these peptides can be like 14 to 20 residues or amino acids long. Okay, and we're going to see here the structure on how that happens. You see, this here is a representation of X-ray crystallography of this antigen on MHC class 2. And you see, this is the hot dog, and those are the buns. Okay, so the ends of the hot dogs can really fall out of the bun. Okay, but then when you look here at your MHC class 1, this thing is really stuck in the center. Okay, so you can't fit a lot of things there. Yeah.
So the ends are only holes on class 1, but not class 2?
Yeah, the ends, because this is only one polypeptide. It goes around, is only one thing. The other one, there are two different polypeptides. So there is some space in between them. That is more flexible to putting in the bigger thing. Okay. So here, again, this is showing you the size of peptide that can be put out there. And again, MHC1 the stuff comes from the cytoplasm, and on MHC2, the stuff is going to be ingested or phagocytosed and then put on the membrane, on the membrane receptor. Okay, so we know that T cells are not going to recognize what is normal. Okay, your native antigens, when you have your cell with the nucleus that has your this is in 3D. Okay, what this means is that the T cells cannot see 3D. Okay, they have to see a piece of that antigen, as I said many times. And also, remember, your T cell is not going to recognize something that is normal in your body, because if the T cell recognizes that, then you're doomed. Then you have autoimmune disease, right? The cells that recognize something that is normal, those cells were killed in the thymus. They underwent the negative selection, so they're not out there in the circulation. Okay. Now, how is it that your T cell is going to see the antigen? Well, it's not going to see the 3D character structure. It's going to see, what is it going to see? Scrambled eggs on a platter. Okay, little pieces of protein stretched out on a plate. That's what the T cells recognize. Okay, B cells can go and recognize the egg, but the T cells, they need the scrambled eggs. Okay, they need stretched out eggs.
B cells also antigen presenting cells?
Yes, they have a MHC class 2, but they are going to interact with the T cells that were not naive, that they were differentiated before, memory T cells. Okay. So here, this is a good thing of this class, this big picture of what's happening with the presentation. Okay, so this is a very simplified version of what is going on, which I like it from osmosis. So what do you have to keep in mind here? This has to do with MHC class 1. Okay, you make MHC class 1 in your endoplasmic reticulum because this membrane is designated to go to the surface of the cell, right? So you make those in the ER. Now, when you have protein turnover in your cytoplasm, you know that proteins are not going to last forever, right? So they're going to be chopped up. When they're ready to be chopped up, they're going to get a tag or a flag or 5 flags of ubiquitin, right? So they get ubiquitinated. They go through a chop up process through a proteasome, and the proteasome chops up into little pieces, and that goes for recycling. In this process, you're going to get samples of those little pieces. They're going to be pushed into the ER. Okay? Now, there is a specific channel of couple of proteins that are going to facilitate the process. Okay, so you call them transporter of antigenic peptides, and TAP for short. Okay, so how do you get chopped up proteins? Well, you start thinking about this guy here was a whole person, and then let's say he gets the arm chopped up. What happens to you if you get your arm chopped up? Where do you go? You go to the ER, right? In this case, you need to be transported. It means that you go tapping to the ER. You know, tap dance? I don't know how to tap dance, but you your arm tap dancing to the ER, and that's how you get to the ER. Okay? In the ER, you are going to have the expression of that MHC class 1 with the beta microglobulin, okay? And it's ready to accept a little piece of protein that was chopped up out there, okay? So this binds to that, but it doesn't just bind, without any help. There is this other protein right here that is anchored to the membrane, to the ER membrane. This protein is called tapazine, okay? I just call it tapazine, okay? So after you do the tapping to the ER, you go through tapazine and you get stuck on the MHC class 1, okay? After that, you're going to make little vesicles, go to Golgi apparatus, and then exocytic vesicles are gonna get stuck in the membrane, and you're showing a piece of the protein. Whatever was inside here is gonna be shown up on the outside of the cell. Okay, simple, right? So just in case I went too quickly through this explanation, I put a little cartoons here showing you what are the processes, right, step by step. So you have your endogenous protein that is tagged by ubiquitination, okay, gets chopped up. Then you are going to get to the ER. Okay, you have to get to the ER. Where is the ER? Okay, it's going to come top 1 and top 2. Okay. Once the chopped up parts are inside of the ER, you are going to have the help of the tapazine to put that on the surface of MHC class 1. Here there are other proteins that are calcium dependent, blah, blah, blah. Just remember tapazine, that's good enough for me, okay? And goes into there, goes into vesicles, gets transported to the outside of the cell. Okay, so once this thing is outside of the cell, if it is a piece of messed up protein like from a cancer cell, then it can be recognized by MHC class 1, okay, because MHC class 1 did not get educated in the thymus about proteins that are transformed in case of cancer cells, right? So if you have a piece of a cancer cell protein that gets exposed here, then your T cytotoxic cell will be able to send a signal to kill the cancer cell, okay? Or if you have a piece of a virus that get exposed out there, the same kind of story. Your T cytotoxic cell will be able to kill the infected cell. Okay. I mentioned that before. Oh, finally, a question. Okay. So this is an easy question because I just mentioned. Here. Cluster of differentiation 8, so that's your CD8, bind to each of the following sites on the MHC, of course, class 1, right? I already gave you half of the answer about the MHC, but hey. Okay, 10 seconds. PDF on the modules, so you should get it. Okay, what's your answer? Is D, is your non-polymorphic region. All right, so what's going to happen here? If you have a cytotoxic T cell that recognizes a piece of virus, this thing is going to, hammer the cell and kill it. If you have a natural killer cell that goes around and doesn't see MHC class 2 over there, I really wanted to get the MHC out, okay? Then the natural killer cell is going to notice that something is wrong with this cell here because that cell should have MHC class 1 on the surface. If it doesn't have, then it's either infected or it's a cancer cell. So natural killer cell should go there and kill the cell, okay? Unless the cell has don't kill me signal. Okay, that could be the case too. But usually if the cell does not have MHC class 1, natural killer cells can go there and kill the cell. Can you think of a case or of a situation where you don't want to kill the cell if the cell doesn't have MHC class 1? Everybody awake? Basically, I'm asking you, which one is the cell that doesn't have MHC class 1? Yeah. Do you want to kill your red blood cells? I hope not. All right? How come the red blood cells don't get killed by natural killer cells? Well, it doesn't have, but that's the job of natural killer cells, to find the cells that don't have MHC class 1. The natural killer cell doesn't know if that cell has a nucleus or not. It doesn't see MHC class 1, it should kill it. How come it doesn't kill red blood cells? Yeah, survival signal. The red blood cells has other ligands that can interact with receptors on the natural killer cells, saying, yes, I know I don't have MHC class 1, but don't kill me, I'm your friend. Oh, I can't remember now. I'm blank. But yeah, there are positive signals and negative signals, and especially for the red blood cells, they do signal the natural killer not to kill them. Yeah. As soon as I step out of here, I remember it. I always like that. Okay. So if you do have survival signals, then then your natural killer cell might spare you. Okay. So here we looked at the endogenous pathway, right? So that could be natural proteins, or it could be something from a virus, right? The virus replicating the cytoplasm. So here you have your exogenous pathway. Okay, so what's happening here? You have a cell that eats up something from the environment. Let's say in this case it's a pathogen, right? It's a bacterium. Or you could have the cell that just ate something, right, from the environment. It doesn't have to be phagocytosing something. It could just be endocytosing something, because the cells eat stuff from the environment, right? So something got inside of the cell. Now you're going to have this process going on here. Remember, you have your phagolysosome. It's going to chop up whatever it ate, right? And you're going to have to put this on the surface of your MHC class 2. Now look at this. MHC class 2 is destined to the cytoplasm of this, to the surface of the cell, so it has to be made inside of the ER, right? You look inside of the ER, and you have all the stuff that is going to the surface of the cell being made there, including MHC class 1. You have MHC class 1, you have MHC class 2. Remember, you have the process with TAP that brings the things from the cytoplasm to stick on MHC class 1, right? Everything is happening at the same time. So you have to have a mechanism that is going to block your cytoplasmic antigen from getting to your MHC class 2. Okay? Otherwise, it's a mess. You don't know which one came from inside, which one came from the outside, right? So what happens here, in the cytoplasm, you have this little piece of protein that is called invariant chain. And that is a plug to the groove of your MHC class 2. Okay, so when these molecules are made, the invariant chain is made, and it goes there and plugs that binding domain. Now, at this neutral pH, the plug stays there. When this protein gets exported into the Golgi apparatus, which is still neutral pH, the plug is still there. When that fuses to your phagolysosome, now the pH is going to change. It's going to be more acidic, okay? So what happens at first you're going to have a partial degradation of your invariant chain. Okay, you're going to have a little clip of the protein that stays there. And yeah, the name of the protein is clip, okay? So a little clip stays there. And then with more acidification, that clip is going to fall off, and a piece of your antigen is going to be able to hop on the groove of your MHC class 2. Okay, so this is the slide showing the overall picture of what is happening here. Okay, so this is your exogenous pathway. because this thing came from the outside. Okay, everybody cool? Okay, so now here you're gonna have your cartoon thing of what is happening. So you have your cell, it is a square cell with a square ER, okay, that is eating something, either endocytosis or phagocytosis. Then, okay, while it's eating, you know, the stuff is happening at the ER. And in the ER, you're going to have the invariant chain that is plugging your MHC class 2 groove. Okay. Okay. Oh, I told you why this is associated, right? You don't want to get your endogenous proteins in there. Okay. So then let's see what happens here. Okay, we talk about phagolysosome. So this is the part that is missing on the overall slide, right? First you have your invariant chain, then it starts to get degraded and you're left only with a little clip. And then the clip, which is right there, is going to be substituted by a piece of your chopped up polypeptide. And that is bigger because you have a bigger groove. Okay. Now, oh, there's something else about the clip thing, okay? The clip doesn't just fall off. It is stuck to the groove, okay? You need a couple of molecules to push the clip out of the groove, okay? So those are the two molecules here. They're called DM and DO. If those molecules are not there, you cannot push the clip out of the groove, okay? So they help to push this out, and they help to load a piece of your peptide in the groove of your MHC2. Okay. Then this gets transported to the surface and you're ready to present it to your helper T cells. Okay. That was the whole story. We have questions on that. Exogenous pathway, endogenous pathway, it's pretty much simple, right? So here we have a little movie showing an antigen presenting cell that is eating something. Here is the eating something, okay? Chopped up the stuff, and then you're gonna have fusion of those vesicles. Okay, so now it totally kept the part of the invariant chain at the clip. So now this goes to the surface and presents it out there. And then you're gonna have your T cell that is going to come and interact with that little piece of protein. Of course, that's your helper T cell, right? And once it interacts with a piece of protein, it's going to upregulate your CD40 ligand that is going to interact with the CD40 on the B cell, and is going to cause other upregulation of other receptor molecules and a double interaction. B cell is sending signal to the T cell is sending signal to the B cell. A lot of cross-talking is going on, right? But remember, your B cell is also an antigen-presenting cell because it really needs to work with the T cell on recognizing the antigen, right? And this is how the B cell shows the antigen to the T cell. Remember the matching, you know, in the nodes, the lymph nodes, or in the spleen. You know? Of course, we're going to see more about that. Okay. Now comes this cool story about cross-presentation. Okay, so at first you think, okay, endogenous pathway goes MHC1, exogenous pathway goes MHC2, but if that is only the case, then you're not going to have a full response of your adaptive system against some types of pathogens, because you are going to need both your helper T cells and your cytotoxic T cells to jump into action. Okay, so let's say, for example, if you have a virus that infects the cell and is replicating in the cytoplasm, right? So a piece of the virus gets chopped off and it gets put into MHC class 1, and then you're going to activate your cytotoxic T cell that goes and kills this cell. But the virus doesn't care because it already replicated. When the cell dies, the virus is going to come up and is going to infect other cells. Right? So you really need to activate your B cells to start producing antibodies to cancel the action of the virus. you need your antibodies to go there and neutralize the virus. You want to tag the virus. You want to totally exterminate the virus, right? And if you only have cytotoxic T cell doing the job, the only thing you're going to exterminate is the infected cell, okay? So the virus can float around and be happy, all right? So in this case, you also have this story of cross-presentation, and the dendritic cells, as you can imagine, they are really, really good at doing this, okay? So the dendritic cell is capable of getting a pathogen that is cytoplasmic and then pushing it to be presented through the exogenous pathway, or vice versa. Okay, so there are several mechanisms that the dendritic cell has in order to do this cross-presentation. Okay. B cells are quite good at it too, but the dendritic cells are really, the best at doing that. Now, so here is saying that you do have a presentation of endogenous in either one or the other. Okay, now let's see how this is possible.
The virus infected cell, but how does it, what does it recognize?
In here?
Yeah, all the way to the left. So you said you have the class 1, so the virus infected cell. What is it actually recognizing on the surface of the MHC on the virus? Is it because for the dendritic cell, I understand that the MHC is going to recognize that, but is it just
Okay, so this cell here is just a regular cell that got infected. Does it have a nucleus?
it's a virus?
No, this whole cell has a nucleus and the virus is inside the cell. Oh, you thought this whole thing was a virus? Oh, no. Yeah, no, yeah, I guess the proportions here are not very good. This thing here is an infected cell. So this is a whole cell. So there's a nucleus in here. On the surface of this cell, you have an MHC class 1 because all nucleated cells do.
I thought the whole thing was a virus and the tag would
Okay, so that was easy. All right, cool. Okay, so here, I'll skip that. All right, so besides your MHC class 1 and your MHC class 2, remember you have those HLA3 genes that I said, oh, forget about them. Oh, now you gotta remember a little bit about them, okay? Because they do make a protein on the surface of certain T cells, and they look like MHCs, but they're not MHCs because they do not recognize proteins. They can actually recognize pieces of lipids, okay, and even some carbohydrates. So basically, you have a lot of types of antigens that can be recognized by those specialized T cells that we're going to talk about next time, okay, after the test, okay. But basically, on the surface of the cell, you're going to have this CD1 molecule. Looks like MHC1 and behaves like MHC2, but it really binds little pieces of fats, okay, little pieces of lipids, okay? So when lipid gets digested, it gets processed and then gets on the CD1 expression on these cells, and then the CD1 expression is going to activate special types of T cells that are usually not the alpha beta receptor T cell. So usually another type of T cell. Okay, so I have a question here saying, okay, most type of antigens can be recognized, right? They can be put on these platters for expression. Bye, see ya. Okay, they can be put on the platter for expression, for a presentation to T cells, right? Now, when you have CD1 molecule with a piece of lipid, is usually is going to show it to gamma delta T cells because those are weird. Okay, they don't have the regular T cell receptor. We're going to meet them next time. Okay, so basically anything can be presented on those cells, except for some things that are not natural, like plastics or metal implants. And the question here is why? Why is it that your adaptive immune system does not recognize these types of implants?
Oh, because your body can't recognize something from when it's from like
No, you're thinking about transplant rejection, yeah.
Yeah.
But this is not a tissue. This is like an implant, like like very popular like breast implants or joint fixing joints implants, metals.
Is it because they only recognize it based on
Oh, but this here is lipid or carbs. They're not peptides. Oh, you want me to tell you why?
Yeah.
Okay, basically because you cannot break down those plastics or those metals. There are no enzymes for that. There are some recombinant bacteria that are able to break down some plastic these days, but the big problem with plastic is because it never degrades, right? I mean, you look at landfills and you have plastics that are going to be there for hundreds of years because you don't have microorganisms or a system that can degrade them, including like in your body. You put plastic in your body and your presenting cells are not going to be able to chop up the plastic and put on the surface of whatever molecule that is, even CD1, right? So you're not going to be activating your adaptive immune system with implants or artificial stuff, okay? That doesn't mean that you cannot build up an immune reaction against implants. Your innate immune system can jump on it and make capsules and get inflammation and all kind of stuff. But you're not going to activate your adaptive immune system. At least that side is okay. All right? So this is one answer for that. Okay, so here is what your CD1 molecule looks like. Okay, so you have a long polypeptide chain that looks like your MHC1. Okay, so one membrane expanding domain. And here you have a huge groove that looks like the groove on MHC2. Okay, and what you bind here is mainly pieces of lipids, but you could also bind some carbohydrates. Okay, so basically this molecule here on antigen presenting cells, it is looking like MHC1, but is acting like MHC2 because of the groove, and is not even binding protein. Okay, so it's binding something else that the cell can find. Okay, so I think that is the story, and that is the last slide. Do you have any questions? Everybody cool? And look at that, we finished 8 minutes early, yay!